Okay, welcome back everyone. So in this session, we're gonna see the tutorial 3 of the GUI. And this tutorial is quite interesting because it has a couple of nice features. The main one is that it includes the reaction diffusion module component of Neuron. So we have some aspects of the model which include some molecular reactions and some diffusion. And I'll show you a diagram of the Rxd, of the reaction diffusion aspects of the model in a bit. The other interesting thing is that the network that we're going to use simulates like a 3-layer cortical network and has some interesting connectivity properties that we mentioned before. For example, things like distance-dependent connectivity and connectivity based on the location of cells. And I will also use the tutorial to illustrate how to save from the GUI to a file, which a couple of people have asked about. And also I will show how to use the interactive Jupyter notebook to explore the model that we are building. Okay, so the first thing we are gonna do is go to the tutorials menu and load tutorial 3A. It says multi-scale network with low IP3. So we're not going to build this model step by step because it would take too long. We've already done some examples of that. So we're just going to explore how the model looks. And so once you load that, you should see first the cell types. Here we have an excitatory and an inhibitory cell type. If you look at the sections, they are like relatively simple models. They have like 6 compartments, 3 apical dendrites, 1 axon and 1 basal dendrite. They have a bunch of ionic channels. This calcium-dependent potassium channel, a couple of calcium channels, 3 types of calcium channels, the sodium, the potassium channel, the HCN channels. Okay, and I think the dendrites have similar ones. Yeah. Okay. And the inhibitory cell type, I think it's a bit simpler. It has 3 sections, soma, axon, and dendrite, and yeah, seems like also a bunch of channels. Okay, so let's see what we have in populations. So here we have 6 populations, and you see they're labeled E2, I2, E4, I4, E5, I5. So they represent 3 layers of cortex: layer 2, layer 4, and layer 5. And in each layer we have 2 populations, an excitatory and an inhibitory. If you click on one of them, we will see we have 10 cells of each. And the I populations are of cell type I, and the E populations of cell type. Okay. So in total, we're going to have 60 cells. In synaptic mechanisms, we have 2 synapses, pretty straightforward: an excitatory synapse and an inhibitory synapse. And in connectivity rules, we see 2 connectivity rules. And this is where it gets a bit more interesting. So we see that we have one that's called E, excitatory, to all. So excitatory cells projecting to all cells. And if we go down this rule, we will see a special thing here. The weight of the synaptic connection, the strength, is not a single value now, but it's using this weird expression here that says 0.04 times post Y norm. And we will see this tomorrow in one of the programmatic tutorials, but essentially NetPyNE has a bunch of built-in variables that you can use to define connectivity. Post Y norm represents the post-synaptic cell Y coordinate in a normalized fashion. So say the cell has a location that is halfway through the cortical network. So maybe this value might be 0.5, assuming the minimum is 0, and the maximum is 1.0. So it normalizes the location of the cell.
And Salvador, sorry, but the expression there could be anything in mathematical form. For example, logarithm is accepted, or
it accepts. Let me just, I think maybe it's worth just showing this right now. So I'll go to the documentation to show this. It accepts many mathematical operations. Not NetPyNE package reference. This is here, functions as strings. So you see the documentation?
yes.
Okay. So I lost it here. So for several of the parameters in NetPyNE, you can, including these ones, weight, delay, probability, convergence, or divergence, you can express them as mathematical expressions. You can include the following things: numerical values, mathematical operators, mathematical functions that are part of this math library. The classical ones are here. You can also include some sort of probabilistic random methods that are provided by Neuron. So you have like different statistical functions, binomial, geometric, log normal, normal distribution. Each of these has different arguments, so you have to check how to use them. But you could, for example, in some examples, we use a uniform distribution to get values within a uniform range of values. And then you have what I was calling the built-in NetPyNE variables, which represent properties of the presynaptic and postsynaptic cell. So for example, you have for the presynaptic cell, the X, Y, and Z location in absolute coordinates in micrometers, the same but normalized from 0 to 1. And the normalization is based on a size of the network that is also defined. You can specify the full size of the network. There's 3 values telling you my circuit is going to be 1,000 micrometers deep and 100 by 100 horizontal. So you can see that.
So in your synapse, did you define it as Y normalized?
I'm sorry?
In your synapse, synaptic mechanisms, did you define it as Y normalized instead of absolute?
In the synapse. So this is what we're talking about is the location of the cell, not the synapse.
I see. Okay.
Yes. So it's the location of the sum of the cell, so to speak.
Quick question, Salvador, because I was trying to use string functions in the GUI earlier. So they do work in the GUI. Just I put mine in quotes and it broke.
Yeah, I don't think you need the quotes. So it interprets it's a string. Yeah.
Oh, we can do way more in the GUI than I thought then. Very cool.
Yes, I'm showing here these network attributes in configuration. This shows the total dimensions of the model that we're building. So the X is 100 micrometers, Y is 500 micrometers, and Z is 100. Okay, so this is the volume of the network. And that is the reference that it will take to normalize these locations of cells. Okay, so you can get the XYZ of the pre and postsynaptic cell, and you can also get things like the distance between the pre and postsynaptic cells that you are connecting at that moment, and it will adapt it to each pair of cells that get connected. So if you have a connectivity rule that affects 1,000 neurons, these values will be calculated for each of the cells involved. So the distance is useful, for example, if you want to have distance-dependent delays. The delay between a connection depends on the distance between the cells. Okay. And then you can actually add arbitrary variables that you can set inside the structured net params, and then you can use them inside these mathematical expressions. Okay, so it gives you a lot of flexibility. You can build very complicated rules with a single line here. Okay, so I go back to connectivity rules. And so this one says 0.04 times post Y norm. So can anyone figure out what this connectivity rule is going to do?
So that's saying that the synaptic connections are stronger at I guess you would say like the top of the Y position, and they get weaker as you get down to the 0 Y position?
Almost, but the opposite. Because the post Y norm is going to be a value from 0 to 1. So the higher values are going to be close to the, so 0 is at the top and 1 is at the bottom. So 1 times 0.04 will be the maximum value and 0 at the top. But yeah, you got the idea.
So you could, so if you were to, for example, change that to Y dist, I think was the way you rate it, that would be that closer neurons have stronger connections?
Yes, exactly.
Okay.
And I just changed it to 1 minus post Y norm, which would be what you said. So that the ones at the top have stronger. And if you did something like Y dist, I think it is. Check. Sorry, dist Y. Dist Y. Then it would be distance-dependent connectivity. So cells that are closer together would have stronger connections.
Okay. I had another question. I think I remember in our last session when we were looking at the connectivity maps, they either had a number, or if there were no connections between neurons, they weren't 0, they were nans. So is there a difference between 0 and a nan for weights if you were to like have zeros instead of nans, or do they function the same?
When you're specifying the connectivity rules, you should use zeros. If you use a nan, it's going to give you an error. But when plotting the connectivity matrix, the ones that do not have any connections might show up as nans. I'm not sure. But in that case, it would be equivalent to a 0. Yes.
Okay.
Yeah, well, we don't. So we had a question of what to do on the connection plotting. If there is no connection, do we show the weight as 0, or do we show it as non-existent? And we settled on non-existent, which is why when there's no cell connection, it's white. If it's 0, it'll be the color 0. You know what I mean? So if there's any color at all, it means there's a connection, even if the weight is 0. So there is some subtle stuff going on there.
Yeah, the reason might be so the the color bar actually is in the range of useful values, maybe, I don't know. Anyway, so we have this mathematical expression for the weight, and the other interesting thing is here in the delay. So we have this other built-in variable, which is the 3D distance between the two cells, divided by the propagation velocity. This is a value that we set arbitrarily. We can set values inside this NetPyNE structure that then we can use in these mathematical expressions. And so we set the propagation velocity to some biological value. I think it was 500 micrometers per second or something like that. And then we will calculate delays in milliseconds based on the distance between the somas of the cells.
And if you want to take into account the distance between the soma and the dendrite, the location of the dendrite, can you do that, or?
You can't at the moment, but it's something that we have. I think we have an issue for that because it's something that would be useful. Okay, so yeah, the pre and post-synaptic conditions, nothing special. So so we have the presynaptic cells conditions for which cells to connect presynaptically were just, I think it disappeared, but it should show the E, because we want to connect all cells of cell type E. So this, I think, was a minor bug because it disappeared from here. And the postsynaptic cell conditions, we are using, just for illustration purposes, this definition, which is we want to connect to all cells within a range of Y values. And in this case, for cells between 50 micrometers and 500 micrometers. And if you remember before, we saw that the full size of the network, the depth of the network was 500 micrometers. So this essentially represents all neurons. Also because layer 2 starts at 50, so we are covering all neurons. I didn't show here in the populations that we're also setting each of these populations at different ranges. So layer 2 is just going from 50 to 150 micrometers. Layer 4 is going from 150 to 300 micrometers. And layer 5, 300. Oh, this one we use normalized coordinates to illustrate. We can also do that. So this one is going from 0.6 to 1. So what would be these values in absolute values?
Yeah, actually, that was kind of my question. I think I confused this with the synaptic mechanism. So why would you have a normalized coordinate for this particular cell population?
Just for illustration purposes, just to show that you can do it.
Okay.
So sometimes in experiments, you get these depths of cells as normalized values. So they use the 0 to 1 normalized coordinate reference with the PIA to white matter being 0 and 1. So that's why we use it here.
All right. Thanks.
Okay, so we have this connected. Yes.
In the synaptic mechanism, when I loaded it, there wasn't any model selected for the mechanisms.
Yes, this seems to be some kind of bug in the representation of the model. I believe the model is still correct. So the underlying Python structure that contains it is still correct, but it's not showing up for some reason.
Okay.
And I can quickly check that. So I was going to show this later on, but you can
an extra T at the end of NetPyNE.
Yeah, you can access the same thing you're seeing in the GUI through the Jupyter Notebook. The structure that contains everything is called NetPyNE Gepetto. NetPyNE because that's the tool, and Gepetto is this actual framework where we're running is based on Gepetto, which is a sort of framework to build graphical interfaces. And then inside NetPyNE Gepetto, we want to access this object called NetParams. So if we check the contents of this, we will see it's of the type NetPyNE specs NetParams object. We can then go inside synaptic mechanism params, and we see that they are actually properly defined. So the excitatory synapse is of X to sin and has the values defined and everything. So it's just an issue with the representation here. We will pass this on to the Metacell guys so they can fix it. Okay. Okay, so the other connectivity rule is this one here. It's called I to E. And this one has another interesting mathematical expression here in the probability of connection now. So here we have 0.3 times exponential minus the 3t distance divided by the probabilistic length constant. So this essentially implements a connectivity rule that is very common in cortex and hippocampus and some other regions, which is an exponentially decaying probability of connections with distance. So cells that are close together are very likely to be connected, have a very high probability of connection, and as you move away, further away, that probability decays exponentially. So that is what is shown here. I think maybe Bill showed an example of this yesterday in the slides. Okay, and then we have some stimulation. In this case, we have this object called background, and I don't think we have seen this in the GUI. This is a NetStim, so it's an artificial spike generator. And for these objects, we can provide the firing rate. So this is going to generate spikes at a rate of 20 Hertz. And you can set here the randomness. So a randomness of value 0 means the spikes are right on target at 20 Hertz. A randomness of 1 means they're all over the place, completely random. So this is a bit random. And then we are connecting that background to both the excitatory populations here. So we target using the excitatory synapse with a weight of 0.02. Here we have another mathematical expression for the delay. we're using this normal distribution. So it's centered with a mean of 5 milliseconds and a variance of 2 milliseconds. And we set the maximum. Oh yeah, so we use max so that the value doesn't go below 1 millisecond. Okay, and we're targeting the excitatory and the inhibitory cells. So I think we can go ahead and just create the network. So creating it itself doesn't take that long. It's mostly the simulation that takes a long time because it has the reaction diffusion built into it as well. And I will show in a bit what the reaction diffusion looks like, but I want to focus a bit on the connectivity right now to start with. Okay, so here we have the network. We can
You can switch to black background too, Salva.
Oh yeah. Black background. Okay. So we have two different types of cells. You can more or less see it here. These are the actually, let's color them to see it more clearly. So I'm gonna go to the coloring thing, and I'm gonna color the E2 population. We can leave this one red. The I2, I'm gonna put blue, bluish. The E4, I'm gonna color pinkish. The I4, green. E5, I ran out of colors. Okay, more or less. So if we go out of the colors, we should see, we can see the different layers now, more or less. Let me just look at it here. So we see at the top here, the layer 2 excitatory cells in red. And we see that they have this sort of lateral dendrite going to one side, the basal dendrite. And we see the blue ones here are the inhibitory cell types, which are kind of weird. They have like a soma and a dendrite, a very thick dendrite. And then we have the same for layer 4 and layer 5. So let's plot the connectivity matrix. So if we go to the sidebar and click on connections plot. Okay. And let's see what we have here. Okay, so let's look just at the first one here at the top, for example. So from population E2, layer 2 excitatory cells, can somebody try to explain what we're seeing here? Why is this square yellow? And the others are bluish.
There's high connectivity between from the layer 2 excitatory to layer 5 excitatory.
You said what was the beginning of the sentence? Huh?
There's just higher connectivity from E2 to E5.
Okay, perfect. And why is that? Based on the rules that we specified? Do you remember more or less?
Oh, because the weights were stronger further away you were. Perfect.
So if you remember, we had this rule that said 0.04 times Y norm. So we're seeing. So that's what we're seeing here, that effect. So we're connecting from excitatory to all other excitatory cells, but with this rule that the deeper cells will have a stronger weight. And that is reflected here in the connectivity matrix. So you see the layer 4 ones, they have slight connectivity. and the ones at the top have a very low connectivity. Okay, so so what are we seeing here? Why is this inhibitory connections to E2 strong, but to E5 weak? And the other way around, like here, the inhibitory from layer 5 to layer 2 is very weak, but the ones from layer 5 to layer 5 are quite strong.
I think we gave this one was based on the absolute Euclidean distance in Y, right?
Yes, exactly.
So they're just closer because they're in the same layer.
Exactly. So this is a rule that is also actually quite common for inhibitory neurons, which is that they are stronger within the layer, within their own region. And so inhibitory connections here are projecting strongly to layer 2 excitatory cells, but weakly to layer 5 excitatory cells. And the other way around, I5 layer 5 inhibitory neurons are projecting strongly to layer 5 cells, but weak to the ones that are far away. Okay, so you see that with a couple of connectivity rules written in a mathematical expression, we got quite an elaborate connectivity matrix, which replicates some of the features of Cortex. Well, this one of the depth was just for illustration purposes, but it's true that we have some examples of connectivity profiles that change with depth, for example. Okay, so let's go back to editing. I think I didn't want to show anything else here. Okay. Okay, so let's open the Python Jupyter Notebook at the bottom. We click on this icon here in the left, and it should open this bar at the bottom. And let's explore what we have again. So NetPyNE Gepetto, NetParams should give you this object. And if we go inside, we haven't yet seen the structure, but I'm going to tell you that there is a structure called Rxd params that contains the reaction diffusion parameters. So if we click on that, we will see that we have a bunch of things inside defined. We can see it more clearly if we do keys, because this is essentially a dictionary. So we see that we have constants, we have regions, species, states, multi-compartment reactions, and rates. And this should sound kind of familiar from the exercise you did yesterday. So what we're going to do is we're going to run the simulation because it takes a while. And while we run it, I will show you what reaction diffusion model is implemented here. Okay, so we're all just going to leave it running. And we want to actually run directly, not just create it. So we can go to the menu model, explore view options, automatic simulation, which means that every time we click the button at the top right, it's gonna create the network and simulate it at the same time. So now it changed, and you can see here at the top right, it says create and simulate network. Okay, so make sure you're not just creating, but both creating and simulating. And again, it's the menu model, explore view options, automatic simulation. You could also actually just click on simulate network. Okay, so I'm gonna click here, and that is gonna take a bit of time. And I'm gonna switch to the here. Okay, so this is the reaction diffusion model that is implemented in the excitatory cells of the network. So the first part here, the first step is.
Your Majesty, you had a pleasant journey out to us? Yes, yes, oh, definitely, yes.
Correct. Craig,
Oh, yeah.
you're muted.
Shit, sorry.
Okay.
Thanks, Greg.
So the first step is simulating a kind of metabotropic glutamate receptor, this mGluR here. This is not literally implemented, but the effects resulting from it are. So this simulates that this metabotropic receptor, metabotropic synapse, is activated, and these synapses usually lead to some secondary messenger cascade reaction where several things happen inside the cell. And so in this case, what we are simulating that happens is that there is an increase in this IP3 molecule in the cytosol inside the cell. So that's step 2, an increase in IP3 in the cytosol. So this, what happens is this leads to the IP3 binding to the endoplasmic reticulum IP3 receptors. So this is based on some experimental data of this real reaction happening. And when the ER IP3 channels open, they release the endoplasmic reticulum calcium from the inside of the endoplasmic reticulum back into the cytosol. So this increases the concentration of calcium in the cytosol inside the cell. And this increase in calcium leads to these calcium-dependent potassium channels opening. So we have here this KCA, which are potassium channels that depend on the concentration of calcium. So the more calcium, the more they open. So more potassium channels open means a flow of potassium inside the cell, which generates an hyperpolarizing potassium current and reduces the cell firing. So overall, we see that we go from increasing the level of IP3 inside the cell to a reduction in the firing of the cell due to this multi-step reaction diffusion cascade. Any questions? Okay. So, okay, so let me see if we have finished the simulation. Right in time. So I hope your simulation also finished, or almost. No?
Not yet.
It should be almost there. So I'm gonna start showing some of the plots. Okay, so I think, yeah, we have changed some feature in the GUI that makes the plots do not scale. So before, when you change the size of the windows, the actual plot compressor, you saw the complete thing.
I took a note of that, Salva. I'm taking notes of all the little things I see so we can talk about them later.
But you can still, scroll up and down and still see the interesting parts. So I'm showing here a couple of things. Let me rearrange this. So we have this one here. So we have the raster plot here in the top left. And we see that, all the populations, they have layer from layer 2, 4, and 5, and 6. And they are all spiking. They have different patterns because we have this random connectivity and random inputs. You can see here in the spike histogram already some kind of the oscillations that are being generated. So you see some peaks here of activity. This is some oscillatory activities actually happening in the network. And you can have a look at the traces. And for the traces, we recorded several things, not just the voltage. So let me show that. So we have the voltage at the top. It's an example, the spikes of one of the excitatory cells. We also have the intracellular calcium concentration, CI, if you remember from yesterday. This was the representation that you had for the intracellular calcium inside the soma. So you see that it is increasing, increasing relatively slowly from -5 to -4. Okay. And we have the extracellular concentration of calcium. This is outside of the cell. So we also have the ability with the reaction diffusion to record not only the concentrations inside the cell, but also outside. And we see that here it's actually decreasing because some of the calcium outside is getting used. Okay. And we have the potassium current. So every time there is a spike, you see these peaks in potassium current. But that's typical of the spikes. So the other thing we can show, which is interesting, is the Lfp signal. So we are actually recording LFPs here at different depths. We have 4 electrodes placed. The top one here is the average, and then we have one in layer 2, one in layer 4, and then one in layer 6 or layer 5. Okay, so you see the Lfp signal, and we can also plot the power spectral density of the Lfp. So we see that there is a peak at around 23 Hertz. Make this a bit wider. So you get the power spectral density for each of the electrodes. This is the one, the average. You can see in layer 2, for example, you have a strong power in the gamma frequencies and a peak here at 25 or so. In layers 5 or layer 4, you have this very strong peak again, 25 Hertz, and less strong frequencies in the gamma power. And in layer 6, mostly just the frequency at 25 Hertz, the peak frequency. And if you look at the spike raster, for example, at the spike histogram, and we count the number of peaks, we should see that it coincides more or less with this peak frequency that we're seeing in the spectrogram. So this is 1 second of simulation. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27. So more or less, that's the peak frequency oscillation that we're seeing in this Lfp spectrogram. We can also plot the spectrogram we're going to plot now. Before was the power spectral density. Okay, and this one is interesting.
You're muted, Salva. Still can't hear you, Salva.
Yeah, sorry, my
There we go.
headphones back. Yes. So we see that in the average electrode, the peak frequency is kind of stable at around 25, 26 Hertz. It actually is interesting because it goes up and down. So we see like over time, the peak frequency changes. In electrode 0 at the top, it's very noisy. So we have all these very high frequencies. And that may be because that's the layer where all the dendrites terminate. So we are recording a lot of synaptic inputs from other cells, and basically all the neuron terminations from many different layers. The electrodes in layer 4 and in layer 5 show a more clear picture of this oscillation frequency, which comes mostly from the spiking activity of the neuron. Okay. And I think, oh yeah, and the last plot that we have here is this Rxd concentration plot, which is showing the concentration of calcium outside of the cell. And we see that for where the cells are located, you see a decrease, slight decrease. Let me get the axis so you can see it. Slight decrease in the concentration of calcium because the cell is using up some of the calcium outside of the cell. So these are locations of different cells. We're looking at the sort of YX view, and you see this slight decrease in the concentration of calcium outside of the cell. Okay.
Yeah, if you show the 2D connection plots, it should line up nicely with those positions.
Yes. A bit crowded here. But, yes, so you more or less see that here at the bottom you see a bunch of neurons which correspond to these ones here. The top there's few more spread out. Here you see one at this location and another one here which correspond to these two guys. Okay, so we are seeing measures at many different scales. So from the concentration of some molecules outside of the cell to the local field potential recorded from a number of cells together, from the activity of a group number of cells. Any questions?
Yeah, Salvador, can you please explain a little bit further how is the LFP obtained?
Sure. So the LFP is the sum of all the membrane currents of all the segments of all the neurons.
Okay.
And it's scaled by the distance, the inverse of the distance to the electrode.
Okay. And there is a mechanism that kind of mix the electrodes as we can observe in experimental data from the volumetric conduction or that kind of things?
Yes, so we're using something that's called the line source approximation. And it assumes this isometric volume. And yeah, there's a number of simplifications. It is possible to modify some of them if you need a more complicated method.
Okay, thank you.
Yes, and just to know that you would still have or can still have these very strong LFP oscillations even if you don't have spiking activity, because it will pick up sub-threshold oscillations of the membrane voltage. So we have examples where we show these LFP oscillations without spiking.
Nice, thank you.
So what's Gramsci plot?
Granger plot is a measure called Granger causality, and it's a measure of how much the activity of one population can be predicted by the activity of another one. So it's a kind of measure of correlation / causation, and it's basically used to try to figure out if the activity of one population results from the activity of another. So if some population is guiding or driving this other population. And I'm not sure if we have it set up here, probably not. Oh, yeah, we have like some default population. So this is actually it's called the spectral Granger causality. So it gives you this measure for different frequencies. So what we see here is that the excitatory population, E2, is essentially driving the inhibitory population, I2, and particularly very strongly at this frequency of 25 Hertz, which is most where all the oscillations are happening.
Thanks.
Okay, so what we're going to do now is go back to the edit. And we're going to change a parameter at the Rxd molecular level. So we're going to change this concentration of IP3 that we saw before in this diagram. So we saw that if we increase the level of IP3, the activity of the network should decrease eventually due to this cascade reaction and the potassium channels opening. So we're going to check. We have to do this programmatically on the Jupyter notebook because this is not yet implemented in the GUI, the reaction diffusion panel to input the parameters. It will be available pretty soon. But for now, what we can do is access it through here. So what we're going to look for is where this IP3 is stored. And let me just remind myself. It's inside species. Okay. So here we can see IP3 is here. We can access that. This is a regular Python dictionary. IP3. So we see that it's inside the cytosol. Its parameter, what is this? The diffusion coefficient, the initial value, the charge, and so on. So what we want to change is the initial value. As we see, it's currently set to 0. And we want to set it to 0.1. We just run this code. I'm going to paste it here on the Ask Tutors channel. And while you're doing that, I'm just going to illustrate that the GUI is synchronized with the Jupyter notebook. So if I do inside net params, the population params, I go to E2. I will see the values here, cell type E, number of cells 10, range, and I can change the number of cells from here. So I want to have 20 instead. And so you see that immediately this is reflected in the GUI here. And if I check here, of course, the value is 20. But if I change back the value here to 10, and I check again, then the value goes back to 10. We have like real-time synchrony between what we do in the Jupyter notebook and what we do in the GUI, which is very useful because sometimes some things are easier from here. Okay, so everybody made this change. Set the IP3 concentration initially to 0.1. Anyone having trouble with this? Okay, so now we can go ahead and again create and simulate the network. So again, this is gonna take a bit. So what I wanted to show is the actual code that is implementing this model. Okay, so we're going to see more about the structure of NetPyNE in the next lesson, but just as a primer here. First thing we have in the code is import from NetPyNE. We import specs, which is the sub-package to define the network. And we create this object called netparams. So netparams is where we're going to define all the parameters of the network. First, we define some sort of general parameters, the size of the network, x, y, and z, this propagation velocity in micrometers per millisecond, and this probabilistic length constant, which is what we use for the probability of connection. We are using this special function that we will see to import cells or load cells from a JSON file, load the cell parameter from a JSON file. You can also do that. Or you can import from a Hope file or a Python file. So we see it's all like a very simple one-line command. And then we're defining the populations. You see very similar structure to what you see in the GUI. It's organized first by cells, then by populations. The name of the population is in the key, and the parameters are inside this dictionary. Layer 2, excitatory. So this is excitatory cell, 10 cells in the range 50 to 150 micrometers. We have the two synapses next, the excitatory and inhibitory with its parameters. And we have the stimulation that we defined before in the GUI. So this is the background input is a net stim with rate 20 Hertz and 0.3 noise. And it is targeting, maybe this long line, but it is targeting both the excitatory and the inhibitory cells. We see that we use this thing called conditions. We specify cell type E or cell type I. We have this delay that depended on a normal distribution. And finally, we have the connectivity rules. We already saw this before. So the conditions of the presynaptic cells is that it's an excitatory cell type. The condition of the postsynaptic cell is that the Y location is between 50 and 500 micrometers. Probability of connection of 0.1. The weight that depends on the Y normalized location, distance, and size. And next, we have all these reaction diffusion parameters. So this is a bit longer of some code here because we have quite a few reactions. We define first some constants, and then we start by defining the regions. We're going to see on Friday, Craig is going to give examples of how to implement reaction diffusion examples that we saw on Monday, that Robert McDougall taught, how to implement those exact same examples in NetPyNE. But just as a quick overview, we can define the regions, like the cytosol, then the plasma reticulum. We can then define the species, like calcium and IP3, and some reactions, multi-compartment reactions in this case, for the different components, the circa pump, this leak, and the IP3 receptors. And finally, we just have what we call the simulation configuration, to specify the duration of the simulation, the integration step, the things that we want to record. We want to record LFPs from different locations. And that is about it. In the last line, this is just some code that we need for the GUI, but usually what you would do is just this line here to create, simulate, and analyze the model. We'll see that in a few minutes. So let's go back to the GUI. Hopefully we have finished. Yes.
Salvador, the file which you showed, can that be directly input as a, can we directly input that to NetPyNE?
Yes.
Okay.
So NetPyNE GUI, yes. And it is essentially what is happening when you do tutorials and click here. It's just a shortcut to import that Python file. So you can do the same by going here, File, Import from Python, and adding that Python file that I just showed.
Thank you.
Okay. So let's see what happened now that we changed the The IP3 level. Oh, I'm not sure if this works. Yes. Okay. So we're actually seeing for a second the old raster, and now we see the new raster. And what we're seeing here is a decrease of activity. You see, after 600 milliseconds or so, the firing rates of the cells are all dying off due to this reaction that we showed. So we increased IP3, and that resulted in a decrease in activity in the network. Just with that small minor change, if we see the LFP, we're going to see the same effect. So you see the amplitude of the LFP is quite strong at the beginning, and it dies off at the end. So we're relating, again, this molecular level IP3 concentration to this more large-scale LFP measure. You can see the cell traces. Okay, so here we will see the intracellular concentration of calcium rapidly peaks once this reaction that we showed takes action. So these IP3 molecules are binding to the IP3 receptors in the endoplasmic reticulum, and the ER is releasing calcium very quickly to the cytosol. So you have this rapid increase in calcium intracellularly, and that results. So this is the extracellular concentration, which decreases initially because it's sucked up, but then because the intracellular concentration is very high, you do not use up so much from the outside. And the amplitude of these potassium currents is higher than before. Okay, so I think we can, quickly, I'm just gonna show back to edit how to export. So if you do export to Python, any model that you build here in the GUI, once you click here, this will generate automatically a file. You can customize the name. I'm not sure if this works. Test. Export. Right.
Yeah, I got the same error while trying to import to
Okay.
Python.
Me too.
So this might be a particular error with this model. I'm not sure. Okay, so since it's not working with this one, I'm just gonna quickly try to see if it's particular specific to this one, because it might have all this reaction diffusion.
Yeah, I think the problem is Rxd, and it's not serializable or whatever. You can't make a JSON out of it.
So yeah, so Rxd is a bit of a special component. So any models that don't have Rxd should work. And I'm just gonna try with the basic tutorial. Gonna load tutorial 1 and try the export to Python. Okay, so that works. And okay, so it exported a file and open. I'm just gonna paste it here for you to see the Python console. This is what I got. So a script generated from the NetPyNE GUI UI, and it has the basic, the network population parameters, the cell parameters, the pyramidal cell, the connectivity, synapses, and so on. Okay, any questions?
Yes, about just a quick question. Can you export the plots as, I don't know, data or something like that in order to plot it in another software or to tweak it a little bit more?
So you can export a couple of things. So you can save the output data of the simulations. You can save it to Python, to Pickle, or to JSON, or to MATLAB format.
Okay.
And you can actually also save the plots both as figures, and you can save the data contained in the plots, so that it's faster to replot them.
Okay, so yeah, that was my question. If I can export the data plots,
yes,
or the data within the plots. Okay.
Yes, both of them. So Joe, I think we'll show examples of that tomorrow, probably.
Okay,
Any
thanks.
questions? Okay, so I think that concludes. What were we? 3:20. Okay, so we'll leave 15-minute break now, and we'll come back at 3:30 for the coffee break for some more NetPyNE programmatic tutorial using the Jupyter Notebook that Joe will run. Thank you,
All right, see you
everyone.
guys in 15.